So there's going to be a bias here. I'm only going to talk about things we've actually done in our own studies. So this might not necessarily be like the way a real animal geneticist would think about these things. But in any case, here we go. So we have a tool that I think we're going to learn more about in the coming days, LD score regression. So there's a particular form of it called stratified LD score regression. And this basically does things like say what percentage of the heritability is due to SNPs on chromosome 6 or SNPs that are near genes of a certain type. And so in order to use this tool for this purpose, you have to have what's called a table of annotations. So the word annotation literally means like a note that you write in the margin next to the main text. So it's kind of a good analogy. I mean, if you think of the genome as the basic text, annotation is some information about extra useful information about a particular base pair or a part of the genome. So here's what the annotation table kind of looks like schematically. You have rows of SNPs. You have tables of these annotations that describe some structural or functional property of the region where that SNP resides. This annotation is particularly interesting. It's called evolutionarily conserved. So the way they derive this, they look at orthologous sequence, because as we were saying earlier, a lot of the genome is shared by common ancestry with primates and mice and even bananas. So I think this particular one comes from a paper in Nature from 2011 where they looked at mammals. So they lined up the orthologous sequence from humans, mice, cows, whales, whatever, and so on. And they looked for regions of the genome that are differentiated in all these different lineages by an unusually low number of substitutions or changes in the T to C or whatever, as you look compared to other parts of the genomes. And so that's why it's called conserved. It doesn't change much across evolution. The inference is that these are regions of the genome that are highly functional. They're important because they're in a gene or they're a regulatory region. And so therefore, mutations that occur in those regions will tend to have phenotypic effects, and those effects will typically be deleterious. And that's why you don't see very many substitutions differentiating different species or SNPs in those regions within any particular species today. DNase1 hypersensitive, maybe I'll explain what that is later. So stratified LD score regression, it has this feature. So there are various histone marks. I think we're going to talk about epigenetics later, actually. But anyway, there's various histone marks, and you can aggregate them at the tissue level. Like if this histone mark is observed in any of the tissues that are in the stuff you can download from Alke's Price's website, then all of those SNPs will get like this central nervous system annotation, and it goes on through all these different kinds of tissue types. OK, so once you have the table of annotations, you put it through the grist of LD score regression. And then one of the outputs is, well, it'll say, like, it'll give you like a regression coefficient, like what is the effect of this annotation on like the chi-square statistic of a SNP that has that annotation. But then it'll also calculate this heritability partition I was talking about. So if 2% of the SNPs in the genome qualify for this evolutionarily conserved annotation, what percentage of the heritability of the trait is explained by SNPs with that annotation? So if it's 2% of SNPs are evolutionarily conserved, but they account for like 20% of the heritability of that trait, we would say that there's tenfold enrichment. The reason why I say evolutionarily conserved is pretty interesting is that it turns out that this always shows strong enrichment, like 8-fold, 11-fold, something like that, apparently regardless of the phenotype. So whether it's years of education, height, schizophrenia, we always see strong enrichment with that particular annotation. So that's pretty interesting. OK. OK, so here's the thing. So stratified LD score regression comes with all these annotations that the developers of that tool assembled, and you'll get them when you download it. But you can apply this tool to any annotation. gathered from any source. So this is what this time around we're doing with this. So here's actually data that was graphed in our paper about years of education last year, and you can see that this basically is referring to this tissue stuff I was talking about. So you can see that CNS is the most, central nervous system is the most enriched tissue. That is SNPs that are in parts of the genome that are associated with histone marks that have been observed in some central nervous system tissue. There are like, such SNPs are roughly 15% of all common SNPs in 1,000 genomes. But they account for roughly 45% of the heritability of years of education, so we have nearly threefold enrichment. But the thing is, there's this other annotation, DNase1 hypersensitive in the fetal brain. What this means is that, so DNase1 is hypersensitive. That means that this is a part of the genome that is easily ligated by this enzyme called DNase1. And that usually it's inferred from that that this is a region where it's called open chromatin. That means that that region of the genome is not like tightly rounded around a histone, which means that any promoters or enhancers that might be there, things like RNA polymerase II and transcription factors can easily just glom on and initiate the transcription of that gene. And so I plugged this in, and I actually did this totally randomly for a workshop about LD square regression I gave back in October, and I was stunned to find that this returns like 11-fold enrichment. So 2% of the SNPs in 1,000 genomes have this annotation, but they account for like 22% of the heritability of years of education. Okay, so those are annotations that have to do with SNPs, but you can also have annotations at different levels, such as annotations of genes. Yeah, there's a question?
Yeah, how is the enrichment measured? Is it percentage increase in R square?
So suppose that 2% of all SNPs have the 1 for that annotation, evolutionarily conserved, say. But if, we might go over the details of this in the coming days, but after you apply LD square regression, and the tool finds that 20% of the heritability of the trait is accounted for by SNPs that have the 1 in that column. So 20 divided by 2 is 10, and then we say 10-fold enrichment.
So it's like a multiplier, so the unit of it is times.
It has no unit because they cancel from numerator and denominator. Yeah. Well, no, that's not right. But it's just what I said. I don't know how to say it differently.
So it's like a
Yeah.
factor, not like percent, not in percent.
Yeah, it's just a factor. Like the SNPs that have the 1 in that column, they account for like X times as much heritability as you would expect based on the sheer number of SNPs that have that annotation. All right. Okay. So those are SNP level annotations. So you can have annotations at the gene level. Suppose that, okay, how do I explain this? Okay, so we have this matrix of genes, right? So this is a toy version of the matrix. The real matrix would have roughly 20,000 rows, 1 for each protein coding gene in the human genome. The columns of this matrix correspond to, we call these gene sets. And so these are genes that are classified according to some database, such as Gene Ontology or Kyoto Encyclopedia of Genes and Genomes, Reactome, as sharing some, both participating in the same biological process or both being, their products being found in the same compartment in the cell. So gene ontology is this whole complicated thing in itself. There's all these curators, and they have to follow all these criteria for classifying all these genes together. In fact, there's a whole book from Springer about just this. The nice thing about Springer is that a lot of universities will give you access to Springer books for free, so you could download that if you want. So anyway, that's what the columns correspond to. And so basically, in this schematic representation, in reality, these are numbers of basically, in this representation, the more the color is toward the red, the more away it is from purple. That means the more likely it is to be a member of this set. So an important point, the original databases, the classifications are binary. So either a gene is said by the curators to be a part of the set or not. One thing we do in this tool that it's called DEPICT is that each gene is assigned a continuous score based on its pattern of co-expression with the original members of the gene set. And so, in our knowledge of biology, there are a lot of errors of omission and probably some errors of commission as well. And so basically, this procedure should give, basically, in fact, it does. We know for a fact it does. It gives us more statistical power to detect what we call enrichment. So what do I mean by that? Oh, I should explain. One other thing first before I go on. So I mentioned Go, reactome, all these other things. It turns out that one of the most useful data sources that we have for this particular application is this thing called the mouse phenome, which was the URL for that was actually in one of the slides. So this is basically studies where genes are knocked out or knocked down in mice, and then you observe the resulting phenotypes. And so this is the basis for classifying genes in the mouse phenome database. And so, for example, decreased brain size. This is a mouse phenome gene set where what the set members have in common is that one of the resulting phenotypes from any of these gene perturbation experiments is that you see that the mouse has a reduction of brain size or brain weight or brain volume. Okay, now let's move on. Okay, so we have two uses to which, well, we have three actually, but I'll just talk about two. We have two uses to which we put to DEPICT. So suppose that this top row here corresponds to one of perhaps many genes that are near a given SNP. So this is a problem we often face. We have a genome-wide significant SNP, but then we're interested in what is the gene through which the effect of this SNP or the causal SNP is mediated. And it might be that there's a gene over here and there's a gene over here and more genes further away. There are cases where the causal SNP is, there are 4 genes in between the causal SNP and the actual causal gene. So we have this question about which gene is it. So the way DEPICT answers this question, suppose that these 2 rows correspond to 2 genes that lie near other significant SNPs. You can imagine that these are perhaps significant SNPs that are on other chromosomes or something like that. Then if it turns out that you have a pattern of data like this, DEPICT will say that the top gene is the gene that it likes. Basically because its memberships in the gene sets is significantly correlated with the memberships of genes that are near other significant SNPs. So it's our belief that causal genes affecting the same phenotype will tend to share annotations and patterns of co-expression. So if that belief is correct, DEPICT is basically built on it, then it will correctly prioritize the genes more often than not. Yeah, there's a question?
I was wondering, in your opinion, how good is proximity of the gene to the SNP a good marker that the SNP is affecting that gene?
It's actually pretty good. I would say that like, for example, I've looked at our latest years of education data, and there's definite correlation in that DEPICT tends to prefer the closest gene, but not always. Yep.
But would that be sort of biased by some genes being longer, particularly sort of neuronal genes? So statistically, you're likely to.
Yes. Yeah. Okay. So that's a great point. So the point is that genes that are highly expressed in the brain tend to be longer. And so if you have GWAS hits and then you define loci around those hits, longer genes are just inherently more likely to intrude into the locus. And so a naive procedure would tend to over-prioritize genes that are highly expressed in the brain. So the way DEPICT deals with this is that instead of parametric statistical tests, it resamples loci from across the genome. And it matches by, it tries to match anyway. There's a huge number of computer science, like incomprehensible stuff that deals with what if you can't find a match and so on. But it tries to match by like how many genes are in the locus. And in the simulations that the developers have run, it's found that this seems to work. When you give DEPICT SNPs plucked at random, or a phenotype where the SNPs are randomly placed across the genome, which is actually exactly the same as just picking SNPs at random, the distribution of p-values is uniform. OK. All right. OK. So that's what we do when we want to pluck out, answer the question, which gene is it? That's where the focus is on the rows. But we can also focus on the columns as well, the gene sets. We can ask, are there any gene sets where the lead SNPs tend to fall near genes that are strong members of the set more often than you would expect it by chance? And basically, what DEPICT will do is just permute these arrows, conditional on matching by how many genes are in the loci and so on. And in the run of permutations, you calculate the sum of the colors each time. If in the run of permutations, the actual sum of the colors in your GWAS data is rarely exceeded, we're going to say that that gene set is significant. OK. All right. So here are some examples of results that have been obtained with DEPICT. So the developers of DEPICT are chiefly Tuna Pers and Ludo Franca. My colleague Tonu Esko likes to call himself the matchmaker who brought together Tuna and Luda so that they could give birth to this brilliant brainchild of theirs. But anyway, Tonu and I, we always share the same reaction when we get new DEPICT results. It's just like you just want to slap your forehead because the results are almost always absurdly good, better than you would have any reason to expect. Like if you look at height, you see all these things like decreased embryo size. This is a mouse phenome. This is a mouse phenome thing. Skeletal system development, another mouse phenome thing. So as I was saying, mouse phenome stuff is almost always at the top of the list. Decreased birth body size, embryonic morphogenesis, cartilage development. As for years of education, you see the mouse phenome category, decreased brain size, absent corpus callosum. This is also basically any gene set that has the word decreased or absent or abnormal is a mouse phenome thing. Abnormal excitatory postsynaptic potential, and so on. OK, I believe that's all.